So, it's always great to be invited to a birthday party, and this is a very special one of a celebration of a station that I know since more than 20 years. It was that guy with the moustache yesterday who was on the slides, who invited me the first time, Serge Poulet, to come here and discuss a paper of mine. So that was the time when people read really papers. And from that moment on, it was 1999, I visited Roscoff, I would say an average once or twice a year. So it's a great pleasure to be here. My group is caring about, I would say, chemical ecology, and chemical ecology as such is a discipline where we try to elucidate what type of chemical signals mediate interactions. And what we are looking for are chemically mediated interactions from microalgae on the one side and on macroalgae from the other side. And this day today, I have brought you two stories from the microalgal field, which illustrate how, I should say, the microalgae use chemicals in order to structure their microbial environment and use chemicals also to communicate not only with each other, but with really the entire microbiome in the sea. So, if we take now the view of a microalgae, and I think in that talk we have to do that from time to time, then this is a cell which is roughly, I would say, 10 to, let's say, 50 micrometers, and it is free floating in the water, or it's crawling around in a biofilm. So you could say that the cell is maintained within its envelope, in case of diatoms, for example, a silicate cell wall. But in the view of a chemist, a cell doesn't end where the cell wall or the cell membranes are, but a cell extends by far beyond its usual range by exuding chemicals. And these chemicals, they can mediate interactions, and they can lead to a communication with the outside world. They can kind of extend the cell. The concept is called the phycosphere, so the cell is building up a sphere of chemicals, and within the sphere of chemicals, you have interaction with, for example, viruses and bacteria. You have interactions with other cells from the same species. They can communicate with each other, like a butterfly or a moth is communicating with another by means of pheromones. Algae, we heard that yesterday, can also communicate by means of pheromones. They can also interact with algae from different species. We call that concept allelochemistry. So allelochemicals can mediate interactions, and we also have the topic of defense chemistry, where I started off here in Roscoff. So what I want to start off is a quite simple, but I would say a pressing question, namely how microalgae can communicate with each other by means of pheromones. And this is a story that I developed together with Wim Wiverman's team in Ghent. It's a story now more than 10 years going, and I want to highlight a couple of ideas to introduce to you the concept of chemical signaling and also maybe our fascination why we want to learn about those chemicals. So let's go to our first model system. It's Seminavis robusta. It's a diatom in a silicified cell wall that is living mainly in biofilms. And if you look at Seminavis robusta and you analyze it, you can see that it has a movement ability. So the cells can move by gliding. They exude like a snail, a slime, and on that slime they glide mainly back and forth. This is what they do. From time to time they take a turn, but that's their, I would say, movement pattern. And this movement pattern will be important for the rest of the story because it's all about movement and searching of diatoms. But before I want to go into the topic of sexuality, I want to introduce to you the life cycle of Seminavis robusta. We heard a lot about life cycles yesterday, and I want to show you basically a simplified picture of it. Seminavis is a diatom, and it's mainly proliferating in mitosis by simply cell division. For doing so, it needs to build up the silicified cell wall. You see here that ornamented cell wall of the diatom, which is kind of a glass house. And to do so, to really be able to divide, what the cells do, is they take up silicate from the outside world, and then they precipitate the silicate to form a biomineral. And this biomineral then shapes the next cell, and by doing so, they build basically the new cell wall inside the old mother cell. And this is quite fascinating, because if you do so, you build your house inside your house, you get smaller. Your house, per definition, has to become smaller. And this is really what happens in diatoms. If they do mitosis again and again and again, the population size in means becomes smaller. And it's a challenge, because if you are too small, you don't have enough space to have your entire biosynthetic machinery in there, and you will die. Nevertheless, diatoms are entirely successful. They contribute basically to a fifth of global photosynthesis. So there has to be a workaround about the dying thing. So you have to be, I would say, have to find a way around that. And if you look at the picture over here, you will see that you have a big cell and a small cell. And this is in fact one clone of diatoms that has undergone rounds of mitosis, and you see here, this is clonally identical, but simply an older and smaller cell. Okay, so what's the workaround? I have been told that after a conference dinner in the first session, you have to be entertaining, so let's talk about sex. And the workaround is, in fact, sex. So they found a way of developing into so-called mating types, plus and minus, like female and male, and those mating types then find themselves, they pair, as you see over here, they shed away their old and small silicate cell wall to form a zygote, to form the oxospores, and then cells of the initial size to undergo rounds and rounds and rounds of mitosis. Okay, so what we are interested in is in that big question mark, the chemistry of sexuality, and also the ecology of sexuality. And together with Wim Wiverman, we characterized in the first place a pheromonal system, because what we assumed is that these cells, this was known already since ages, that these cells have to find themselves, because otherwise, if it would be based on serendipity, they would never meet and would be extinct as well. So we discovered a system of really nice and orchestrated interactions, and this is mediated by pheromones. The so-called attracted mating type, or mating type plus, releases a signal once it's small enough to have sex. So once the cell size is reduced and the cell has a sexual capability, it releases a signal that constantly signals, I am here, I'm small, I want to have sex. This is really the message that it switches on. It's a highly regulated thing, because once the so-called attracting mating type receives that signal, it changes its physiology, and it produces a signal, hey, if you follow that trail, what you will find is me, and I'm also small, and I also want to have sex. So basically, they communicate in a manner with two, at least, chemical signals. And we wanted to learn about the first chemical signal, which is the follow that trail signal, so the attraction pheromone. This is an experiment that we then designed in the lab. We used a culture of the mating type that releases that attraction pheromone and grew it monoclonally. So there was no one else around, no one else signaling, I am here, I want to have sex. So they don't produce the pheromone. This is that control culture. And then we split that culture into two, and we used a mating culture where the I am here pheromone is in place. And we used a drop of this culture to trigger now pheromone production in those guys. They are still alone, but they receive the I am here signal by our pipettes. So not by their sexual partner, but by pipetting in the signal. And in that moment, they believe, I would say, that someone else is around who wants to be mating, and what they do is they then release the attraction pheromone. We can extract that attraction pheromone using solid phase extraction, and we can use the plastic beads where we loaded the pheromone on in order to do a bioassay. What you see here is the plastic bead that we loaded with the pheromone, and this is 10 times accelerated. The picture of the moving seminavis who is desperately trying to mate with a plastic bead. Poor guys. Well, you have to give sacrifices for science. I think this video doesn't need much more explanation because it's so obvious. You place one of those plastic beads in and then all those guys gather around. So it's I would say, clear proof that we extracted the pheromone. Now we want to get the structure, down to the structure of the pheromone, and what we do is use metabolomics methods in order to investigate differences between those two cultures, the calling one and the control cultures. We do that by statistical analysis, so we look for significantly upregulated metabolites, and I would say I wouldn't have believed it myself, but in fact, the statistically most upregulated metabolite that the computer pointed us to was in fact the pheromone. So we never believed that. The big challenge in chemistry, if you look for chemical compounds that mediate interactions, is that you have, like here in that chromatogram, hundreds of compounds that are exuded into the water. That's what a cell does. It's not a closed shop, but it releases compounds into the water. Hundreds and hundreds of them. And there were decades of efforts in order to isolate diatom pheromones by trying to purify fraction by fraction of these compounds. And it doesn't work. You lose your activity. The pheromones are highly active. You have very minor amounts, and that doesn't work. But comparative metabolomics is an art, because what you want to do is you want to throw 99% of the information into the trash bin. And this is really what we do. We consider that all the compounds that are in the control and in our treatment cannot be the pheromones. So we ask the computer to eliminate those and to show us only those that are somehow triggered in a sexual event. And in fact, the compound that we isolated was diprolin, a dimer of 2 prolin molecules. We could synthesize it, do the structural elucidations, that's what chemists do. And we could use it then for experiments to see if we in fact have the true attraction pheromone in there. We again used our plastic beads, but in this case we didn't load them with the natural extract, but we loaded them with a synthetic pheromone. And we found a response curve that is quite clearly a receptor indicating a receptor mediating response. So what you see below is a logarithmic scale of concentration of the pheromone. And here you see the response, the attractiveness of the beads. And like in any receptor-mediated interactions, we find basically 3 orders of magnitude in terms of dilution, where the pheromone is active and highly attractive. We have a threshold concentration, which fits quite nicely with the concentration in nature. And we have, as in any receptor interaction, a saturation, because the receptor is then loaded with the compounds. It cannot get rid of them anymore, like in our nose. If we smell a compound for too long, we don't smell it anymore. The same here, so if the concentration is too high, the algae are not attracted anymore. So we have the pheromone in hand, and we can now use manipulations and do a lot of investigation. Wim Wevermann's group is using it to track the sex locus in the genome. We use it to analyze, for example, movement patterns. So what we can do is we can do video recording of the cells that are attracted to the beads. We find that the cell movement is strongly accelerated when the cells recognize the pheromone. It's a so-called chemokinesis. I speed up because then you increase your chance to meet a partner. Sure, if you work slow, you will not encounter as many people as if you run around in the room. And the same is here done by the diatoms. And the other thing that they do is they make a quite smart move. If the flower bouquet would be my mating partner, and I only have the possibility to go back and forth, then I would not meet it. But if I can integrate the concentration of the pheromone and prolong my forward movement in comparison to my backward movement, 3 steps beyond, then sooner or later I will find my mating partner, and That's it. So this is really a quite nice way of mediating sexual interaction, and I am really fascinated by the way how the diatoms respond. So that would be now the story of the attraction pheromone. But now I come to, I would say, result that is derived from our studies and that is also connected to the physiology of the diatoms. I told you they need silicate to build up their cell walls. And what you see here are these nicely ornamented pictures of diatom cell walls. And if you need silicate, silicate is also depleting in the medium. Silicate is also limiting in the outside world if you have lots of diatoms. And what we found is that motility is not only connected to sex, but motility is also connected to silicate concentrations. So if the cells get hungry, they again speed up. Why do they do that? Clearly enough, they are searching for new food sources, for new patches of silicate. And what we did is we derived silicate-loaded beads and added them to the cells. And in the next video, you will see on the left-hand side a silicate-loaded bead, on the right-hand side a control bead. Look at the movement patterns. I will show the video then once more and explain what we interpret into them. So left, silicate loaded, right, controlled. Okay, so once more, you have the cells over here. To take that, sorry. You have the cells over here, and they gather around the silicate-loaded bead. First, they speed up, they go back and forth, and if they recognize that the concentration of silicate is highest around the bead, they settle down and do their thing, they proliferate. So basically, again, we have an attraction, but this time an attraction, and this was really new, to an inorganic nutrient. So we have an attraction on the one hand to a pheromone and the other hand to a nutrient. So what we asked then is really a quite simple question. Can we have a behavioral ecology of diatom cells? We have two responses that both are an attractive response to food and to a pheromone. So we can ask now a cell, do you want to have food or sex? An everyday question, and this is really, I would say, quite nicely. Diatoms, they are unicells. They don't have a brain. They cannot compute. So we were really wondering, can they somehow prioritize food or sex? So what we did is we used cells of different sizes, the large cells that simply divide, and they are not responsive to the pheromone. We know that. They are responsive to silicate. And then we have cells that are smaller. They just cross the sexual size threshold and they are ready for sex. So I'm a father of four. I think it's quite counterintuitive for me to say, hey, well, you're not small enough to have sex. But we know that multicellularity has evolved a couple of times, so maybe they have thought of a different strategy and you have to be small to have sex in contrast to us. Okay, so we have the cells just below the sexual size threshold and then the cells that are completely desperate, small. If they continue division, they will die. So we derived an experiment where we offered pheromone-loaded beads and silicate-loaded beads and looked at the responses. And we see that if the cells are kind of just ready for sex, everything has to be in place. They have to be saturated with silicate, the I am here signal has to be there, and then, oh yeah, well, we go to find our mating partner. If something is not right, the attraction pheromone, the induction pheromone is not there, or if they are hungry for silicate, they simply do not move. And the situation is completely different when the cells are tiny, 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 and try to, or have to do sexual reproduction. Then they throw overboard all their control mechanisms, and no matter what, they are attracted to the pheromone. So it's a quite, I would say, intriguing story, because we have now unicells that do a behavioral response. Okay. So I have a couple of minutes left, and I will use those couple of minutes in order to make the story a bit more complex. The idea is that algae are not only alone, like in the example that I showed you, but they are always in interaction. 5 minutes, yeah, I will make them. So what we asked around is what would happen if the algae are interacting chemically by means of chemical compounds, and we wanted to do that in investigation now by using a setup where the algae are in chemical contact. They can exchange chemicals, but they are physically separated. So we separate those cells by using a membrane. And what we found is that if you are an algae and you're not alone, like in the pheromone example, but you're in